Dear viewers, today we are going to discuss about identification and preservation of the weeds. I am Dr. I.C. Borua, Principal Scientist in AICRP and Weed Management in Department of Agronomy, Assam Agriculture University, Jorhat here. Getting a name for a plant, it is identification for us in weed science, getting a name. So name may be of different types in our local languages, in Assamese, in English or in other languages, as well as botanical name also. So that is why though we follow an universally accepted name, and this is called a botanical name or scientific name. So ultimate goal of identification is to get that scientific name. Then what is the scientific name then? In scientific name, I think you know that it is a binomial nomenclature. There are two words are there. First one represents the genus, say Abelmoschus. Second word is species epithet. When species epithet is joined, it's clubbed with the genus name, only then it represents the species name. You see, what is esculenta? Or it is means edible. And this word, when we joined with Abelmoschus, means genus name Abelmoschus and species epithet become esculenta, then it represents the Abelmoschus esculenta, that means bandy plant. But if we use this esculenta with other genus, it will represent other name. For identification, what happened? We used to collect our plants. First of all, we characterize. What are the important characters? We go through it, we characterize, and then we match the characters with published literature. We call it a flora with literature, and then finally we ascertain that, oh yeah, this is pen on literature, it is okay. But visually we have to confirm. Then we have to bring your specimen to a reputed laboratory, and that laboratory is known as a herbarium. Okay, we have a Central National Herbarium in Howrah. Nearly 2.5 million herbarium samples or seeds are deposited there. So we can prepare our herbarium seeds by following the protocol. Then we can send to this type of reputed herbarium by post also, and there it is, the name is, when the name is found, then they will inform you that your specimen is this. So herbarium matching is the final stage. Before that, we have to characterize our specimen, compare with the literature, and then we have to finally match with the existing herbarium seeds. So now we'll demonstrate how the herbarium is prepared. First of all, we have to remember that our size is limited. It is 42 centimeter in length and 28 centimeter in breadth. So our specimen should come within this frame. This is a common chase, Cyperus iria. So it is length is almost it is below 42 centimeter. That is, it is no problem. We can collect these things. So let us collect this one first of all. So when we collect a weed, first of all we have to fix one tag. This is the tag. So what we'll write there? We'll write abbreviated name of the collector and collection number. Need not to remember what was the last collection number. Every day we'll start with 1, and then we'll write down the date. What description we should write? First of all, you see, when we fix this plant, we can read the characters from the specimen. So that is why now we'll record which characters are not being preserved. First of all, whether it is deep rooted or shallow rooted. Okay, it is deep rooted or shallow rooted, and it is extremely shallow rooted. Therefore, you write down shallow rooted. Then habit, it is herb. Okay, but when we cut the twig, sometimes we used to cut the twig. Then by seeing this twig, we cannot ascertain whether it is a herb, shrub or tree or climber. So then we'll write the habit also. So therefore you have to write herb. This is a herb. Okay. You see the stem, triangular, all these characters we can read by seeing the specimen. The color of the flower may not be preserved because during pressing and drying it may lost. So that is why color of the flower. So there is no flower, it is spikelet. You write down the spikelet. It is yellowish green. Yellowish green. So this much is required, and out of that we have to write down the local name and if any use is there. Say it is a bushy one. Okay, it is a bigger one and it is larger than 42 centimeter. Then what we'll do, we'll cut a twig, this one. Then we'll fix one tag. This is my collection number 2. First of all, we write down abbreviated name, ICB, my abbreviated name. Then collection, it is second collection, I'll write 2 and then I'll write the date. Now what will be written here, description, it is not exactly a herb, it is a undershrub. You write down undershrub. And then just when you try to pull it up, then it is a deep rooted. Okay, you write down deep rooted. Color of the flower is very, very important. You write down the color of the flower, it is yellow, and sometimes, many a times what happens, the petals fall off. Therefore, we write down number of petals 4. There are 2 names. One is Pani Jatuka, comma, Bon Long. Within bracket, you write down Assamese. You say this is a grass, shallow rooted. So now you write down shallow rooted herb. So here, if we cut the twig, only the inflorescence and flag will come and the rest of the leaf will not come. So 2 things we can do. We can cut into number of pieces. Okay, and if we cut into number of pieces, then for each piece we have to fix individual tag and we'll write the same collection number. Otherwise, since it is a grass, lean and thin stem is there, we can fold it to fix in the herbarium. We can fold it, easily fold like reverse V or sometimes like M. Without flower, it is very difficult for identification for many plants. These are very common plants, of course, this is different thing, but we should not collect without flower. We should collect with the flower. You see, this is the climber. It may be longer, no problem, but we can roll it like that to keep within the frame. So this is all about the collection, what to be collected. How much to be collected? This is another important thing. You see, after collection, we'll press it, dry it, and during pressing and drying, it will take sometimes couple of months. And we'll continue our collection, and during pressing and drying, some specimen may get damaged, or sometimes it may be lost even. So that is why better it is advisable to collect 3 samples of each. For each one, you can collect at least 3. So we have collected 1, 2 and 3. So after pressing and drying, we'll select the best one for our herbarium. And second one, we can keep ourselves, because this main herbarium what we are going to prepare, we have to submit somewhere, either as an assignment or to a herbarium laboratory, we will have to submit. So therefore, another one, after identification, we'll keep ourselves as our reference, because after identification is a very tough process. So during the process, we will get the name, and if I can keep this one with me, then it will be for me, it is a future reference. So, and third one will be surplus one. You can exchange with your friend or anybody else, or you can later on you can discard one. But it is advisable to collect at least 3 samples from the field at a time of collection. So what we collected in the field, now these are to be pressed and dried. So these are pressed in the whole in the fold of old newspapers. So why we use old newspaper? It is the cheapest blotting paper and very useful. Otherwise, you can use blotting paper also. So inside the fold of newspapers, we keep it. Then in the press, we press it. Say you don't have this type of press. This is made up of 2 wooden boards. So anyway, no way, you can keep the specimens one by one, keep some blank newspapers over there, and give some general press with the help of some bricks or big books. But remember one thing, you have to change this blotting paper every day, at least for 7 days. After that, you can keep it for longer period, because our humid weather is responsible for damaging our specimen. So if you don't change the paper, so what will happen, your specimen will be fungal attack and it will be damaged. During the second and third changing, when you change the papers, then you arrange your specimen beautifully over the paper so that overlapping is minimized, and some remember, some leaf should face up and some leaf should face down, and arrange beautifully and press it for at least for 1 month. For first 7 days, you have to change the papers, and then you can keep it for longer period. So when your specimen will be properly dried up, then it is mounted. Mounted means it is glued, And then what we use Fevicol or Vemicol, this type of glue you can use, and with the help of this Fevicol we'll fix our specimen over a mounting board. Now question is, what is the mounting board? So this is the mounting board. You see, it is a universally accepted size of that mounting board is 42 centimeter in length, 28 centimeter in breadth, and its right hand bottom corner is reserved for the label. And there we will fix our specimens in a longitudinal way, not in transverse way. And when the specimen become bigger, then we can fold it like this, or sometimes we can make it W. And I told you that if it is a climber, then you can make it a circular way, we can arrange. So it will be fixed, and after mounting, when we use the glue to fix the specimen, this is called mounting. After that, again we have to keep in the press, covering with newspaper. We should keep it at press for at least one night so that it is properly fixed. So when it is ready now, then what will happen? These specimens, some stiffer organs are stitched. Okay, these are stitched, and then when it is stitched, then it will be ready finally. Then I will show you. See, so these are some old herbarium sheets prepared by students only. You see, it is beautifully stitched. And then what will we do? We classify based on the genus and family names. After identification, and this is called herbarium cabinet, and there we keep genus wise and family wise we keep, so that anybody can find it out. And ultimately the genus and families are arranged alphabetically so that you can identify. And what is the use of these? Say here the specimens are there of one genus or one species collected from different places from different times. And if you observe, if you see these variations, what are the morphological variations in the seeds, then you can write about the descriptions and variations of all the seeds. So this is the use of the herbarium seeds. And another use, you can consult this herbarium for identification of your unknown specimen. What we have seen, this is dry preservation. Now many specimens are there which are very tiny or succulent. Many a times we preserve in liquid preservatives, and one of the best liquid preservative is FAA solution. So there are two ways we prepare FAA solution. If we keep the specimens for a longer period, then what we use? We don't use water. And if it is a short duration for 2-3 months or 6 months like that, then we can use water also. So what is this? This FAA solution means formaldehyde. 10 ml formaldehyde we use in both the solutions, 10 ml formaldehyde. When we use water, we use 50 ml of ethyl alcohol. But if it is very tiny one and succulent plant, then we use don't use water. Then it is 85 ml ethyl alcohol. Okay, and when we use water, it is 50 plus 35. 50 ml ethyl alcohol plus 35 ml distilled water. And third one is the acetic acid, glacial acetic acid. This is the glacial acetic acid, 5 ml of glacial acetic acid used. So what is the solution now? Formaldehyde 10 ml, acetic acid 5 ml, and alcohol either 50 and 35 ml water, or all 85 ml ethyl alcohol. Now we are using a very tiny plant. This is Peperomia pellucida, a very common weed. In Assamese we call as a damdeuka. And now we'll keep one specimen there. So this is a process of liquid preservation. This is the best solution. It is called FAA solution. Anyway, thank you.
Hello viewers, myself Dr. Huntara Kolita. I'm working as an assistant professor, Department of Agronomy, Assam Agriculture University, Jorhat. Dear viewers, today in this video we are going to demonstrate the method of application of biofertilizers. Before going to explain or demonstrate about the method of application of biofertilizer, first of all, let us know about biofertilizer and its different types. Biofertilizers are biologically active products or microbial inoculants consisting of living cells or latent cells of efficient strains of microorganisms, which may help in increasing crop productivity by way of helping either directly or indirectly in the enrichment of soil fertility through the biological nitrogen fixation, solubilization, and absorption of insoluble phosphorus, stimulating growth or help in decomposition of plant residues. Use of biofertilizers is one of the important components of integrated nutrient management, as they are cost-effective and renewable source of plant nutrients that supplement the chemical fertilizers for sustainable agriculture. It can replace 20 to 50% of nitrogenous fertilizer and 15 to 25% of phosphatic fertilizers. Generally, there are two types of biofertilizers. First one is nitrogen fixing biofertilizers. It includes rhizobium, that is used for leguminous crop, Azotobacter, Azospirillum, Azolla, blue-green algae, that is used for cereal crops. And second type of biofertilizer is phosphate mobilizing biofertilizers. This group includes phosphate solubilizing microorganisms and phosphate absorber. Several soil bacteria and fungi like species of Pseudomonas, Bacillus, Penicillium, Aspergillus, etc. are phosphate solubilizing microorganisms. Today, we will demonstrate the method of application of nitrogen fixing biofertilizer and phosphate solubilizing biofertilizers. There are mainly three methods of application of biofertilizers. First one, seed treatment. Second one, seedling root dip treatment. And third, soil treatment. Now we will demonstrate each method one by one. Today we will demonstrate seed treatment of green gram. For seed inoculation of 1 kg of green gram, we have to take about 50 gram of rhizobial culture, 50 gram of PSB and 10% jaggery solution. Here we have taken 10 gram of jaggery in 100 ml of water. Jaggery acts as substrate to the microorganisms. It also helps in proper coating of biofertilizer with seeds. First of all, we have to mix this solution well to prepare the slurry. Here is the slurry. Then this slurry is mixed thoroughly by hands with seeds of the crop, so that all seeds are uniformly and equally coated with the product. Then coated seeds are sprayed in the polythene sheet and allowed to dry under shade. These treated seeds have to be sown as soon as possible. Seedling root dip treatment is followed for transplanted crop, mainly in rice and vegetables. Today, we will demonstrate the method of seedling root dip treatment in transplanted rice. A bed is prepared in the main field to accommodate seedlings required for 1 hectare of area, and that is filled with 2 inches of water. Here, for demonstration, we are taking rice seedlings for a small area. For seedling root dip treatment, the roots of the seedling must be washed off to avoid soil from the nursery for proper exposure of the roots to the biofertilizers. Generally, for seedling to transplant in 1 hectare of area, about 4 to 5 kg each of Azospirillum and PSB is thoroughly mixed with water in the bed to prepare the slurry for root dip treatment. Here, for 100 meter square area, we are taking 40 to 50 gram each of Azospirillum and PSB. Then the rice seedling should be dipped in the slurry for 8 to 12 hours overnight and then transplanted on the next day morning. Today we will demonstrate soil treatment of biofertilizers, for which we need compost and PSB and Azospirillum as a biofertilizers. Generally, for 1 hectare of area, 4 to 5 kg each of Azospirillum and PSB should be mixed in 4 to 5 liter of water separately, and both the suspension should be sprinkled over 100 kg of compost or FY. For demonstration purpose, we are taking only 5 kg of compost along with 250 gram of PSB is mixed with water to make the slurry, and 250 gram of azospirillum is mixed with 250 ml of water to mix the another slurry. We have to mix this slurry properly. We have to mix this slurry separately to the compost and mix it well. If soil treatment is done for large area, then we have to divide this compost into different heaps, 2 or 3 numbers of heaps, and then we mix the biofertilizer for easy handling and proper mixing of the biofertilizers. After mixing, the compost and biofertilizer mixer should be left overnight for incubation before application in the soil. After 12 hours of incubation, different heaps are mixed together. Now we'll mix 1.25 kg of lime along with this biofertilizer mixed compost. Generally for acidic soil, about 25 kg of lime is used for 100 kg of compost. As we have taken only 5 kg of biofertilizer mixed compost, so we take only 1.25 kg of lime and mixed it properly. Then this mixture is broadcasted evenly in the moist soil and mixed with the soil just before sowing. Dear viewers, I think you like the video, and if you have any query, you can contact us. Namaskar.